In this video, we're going to do a quick introduction to the different components that you'll find in blood. So just for a very brief overview, when you take a blood sample and centrifuge it, so this is a tube that was put in a centrifuge, spins around in a circle really, really fast, and things that are more dense will sink to the bottom of the tube. And red blood cells, or erythrocytes, are by far the most dense. They're full of hemoglobin and iron, and they sink straight to the bottom. The top is plasma. That's the liquid portion of your blood that has dissolved nutrients and minerals and dissolved proteins in it. And then in the middle, you have this thin little layer that looks white, almost creamy colored. And this is sometimes called a buffy coat. That's just white blood cells, right? And maybe platelets to some degree. Right? So these percentages, in a typical person, somewhere between 50 to 60% of your blood is typically plasma, and somewhere between 40 to 50% is typically red blood cells. White blood cells take up a very small percentage, and remember, the reason for that is white blood cells mostly live in the lymphatic system. Lymph nodes, spleen, thymus in younger children. Not that many of them circulate because it would be kind of a waste of effort for your heart to be circulating all of these white blood cells when we really can just leave them sit in lymph nodes and wait for the infection to come to them. Right. And when we quantify all this, we use a number called hematocrit. Hematocrit is really just the percentage of your blood volume that's made up of red blood cells. And so in this example, this person would have a hematocrit of 45%, right? Because that's how much of this total volume was red blood cells. Okay, so within a whole blood sample, if you put it on a microscope and stain it, you'll see a handful of different things. The first is the vast majority of the cells in your sample are going to be red blood cells. They are sort of pale pink colored. Some of them almost look hollow because they're very concave. They're not really hollow in the middle. The membrane is just very thin, so light passes through it pretty easily. Any of these larger cells that have a nucleus, right? The purple staining is a DNA stain, that's a nucleus. Those are all white blood cells. White blood cells absolutely have to have a nucleus because they need to be able to divide. Right? In red blood cells, we get rid of the nucleus because DNA doesn't carry oxygen, and it would be a waste of volume in the cell to just pump those chromosomes around a person's body all day long. Your heart has better things to do. All right, so white blood cells are large and nucleated. Red blood cells are smaller and anucleated. And lastly, platelets are just fragments of cells called megakaryocytes that fracture into tiny little pieces. And so they look like little clumps. They're like bits and pieces of cells, but they're not really fully functional living cells anymore. Right? And those platelets, ultimately, they're going to be part of the clotting pathway. So when those megakaryocytes break into platelets, they start off as inactivated platelets that are not sticky at all. Right? They're just floating around your blood, doing their own thing. And something has to activate those platelets to turn on this coagulation cascade. And coagulation is really just a mixture of two things. It's the activation of platelets to make them become sticky and bind to each other, and the activation of a protein called fibrin. Those two things together, fibrin plus platelets, cause a clot, right? And the clot winds up being a bunch of those fibrin strands and chunks of platelet that get bound up in a pile of red blood cells, and that's what actually stops the bleeding. Okay, so in this video, we're not going to go through the details of the clotting cascade. I'll record those details separately in a different video for people that need them. But just know that you have many, many, many regulated steps in the clotting pathway, and there are a lot of available drug targets. So if you inhibit any one of these steps in the clotting process, going from inactivated platelets and fibrin to the activated form, like heparin, warfarin, aspirin, they all have different mechanisms, but the thing they have in common is they reduce clotting, which is helpful for people that have blood clots that need dissolved, or patients going into major surgeries where we want to prevent systemic clotting as the surgeon cuts through their tissue. Okay, so moving on from platelets and red blood cells, we're mostly concerned with white blood cells in this section, and I wanted to give a brief background on where they come from. So all white blood cells are initially derived from bone marrow, and you have cells called hematopoietic stem cells, right? Which really just means they're stem cells that can divide and potentially become any different white blood cell or red blood cell type for that matter, right? Every time you see a fork in the road, we call that process differentiation. And differentiation, big picture, really just means that the cell is becoming more and more specified, more mature, getting closer to its final destination of being a mature functional cell. And along the way, we have multiple splits, right? So these myeloid progenitor cells can become either red blood cells or mast cells or neutrophils or eosinophils, right? Or basophils. And the lymphoid progenitor cell, right? T cells and B cells are really just short for T lymphocytes and B lymphocytes, right? And those are all derived from lymphoid progenitors. And every step along the way, we get more and more specified, right? Closer to a final function, right? So these different white blood cell types, right? Everything that you're seeing here, these are our functional cells of the immune system. And this series is going to focus mostly on what's happening on the lymphocyte side of things because that's the most interesting. But these other cell types have important functions too, right? These guys, monocytes, macrophages, neutrophils, eosinophils, you can mostly lump these together as being phagocytic cells. And they might attack slightly different types of pathogens and be found in slightly different locations. But phagocytosis is their primary goal. Basophils are primarily involved in the inflammation response. So they release a lot of those cytokines that trigger inflammation and itching and swelling to make a inflammation response actually happen. All right. Now, where we're going to spend most of our time is on lymphocytes, right? And the big picture story is B lymphocytes or B cells are primarily responsible for producing antibodies. T cells have a handful of different subtypes. There are actually more than 3, but we're going to keep it fairly straightforward here. There are inflammatory T cells that trigger the immune response and attract these phagocytic cells to the site of an infection. There are cytotoxic T cells. These are sometimes called CD8 T cells because that's the receptor they have. They kill cells infected by viruses and sometimes cancer cells as well. And then you've also got helper T cells, which are sometimes called CD4 T cells. And their job is sort of to be like a master coordinator of the immune system and stimulate other immune cells to amplify their responses. All right. So to kick all this off, I thought it would be helpful to do a big picture overview of what's going on in the immune system. All right. The beginning of this, some sort of pathogen, right? A virus, a bacterium, whatever it might be, has to enter the body. And the term antigen, remember, an antigen is just a molecule on the surface of the pathogen that your immune system can detect, right? So this could be a cell membrane protein of a bacterium or an envelope or capsid protein in a virus. And this macrophage or any other phagocytic cell, they happen to be showing a macrophage, is going to start digesting and phagocytizing that pathogen. Right? This is where those pathogen-associated molecular patterns would come in because that's what the macrophage is recognizing. Okay? Once that pathogen is broken down into little bits, that macrophage is going to present parts of the pathogen and show them off to immune cells, particularly T cells. Right? Now, that showing off or presenting of the antigen, we call this cell now an antigen presenting cell, or APC, right? So the macrophage, once it starts showing off the antigen, becomes an antigen presenting cell, and we're looking for T lymphocytes that recognize it. Okay, now depending on the details, that T cell might be a killer T cell, which is the same thing as saying a cytotoxic T cell or a CD8 positive T cell, right? What these things do is they look for antigens that are inside of human cells. So when we say infected body cells, that really means cells that are either infected with a virus, or it can also mean cancer cells. If the cancer cell has a mutation that makes it look foreign, they can also be killed by CD8 cytotoxic T cells. And the mechanism is just these killer T cells release digestive enzymes, reactive oxygen species, and they can also recruit macrophages that help out in the job to destroy that virus-infected or cancer cell. Okay, the second thing that can happen is these can be recognized by a CD4 or helper T cell, right? And if it's a helper T cell, their job is just to stimulate and activate other cells in the immune system, like B lymphocytes or B cells. The B cell's job is to produce antibodies, right? So on the surface of the B cell, these molecules that you're seeing right here, these little Y-shaped guys, those are your antibodies. The little pink dot, I think, is supposed to represent the pathogen, the bacteria or the virus. And what you expect to happen is, if this B lymphocyte recognizes the pathogen and it gets stimulation from a helper T cell, it's going to start dividing or proliferating. And the end result from that B cell dividing is you get two different things. You get plasma cells, which, as their name suggests, secrete antibodies out into the plasma. You expect those antibodies to float around and start binding to the virus or the bacterium wherever it happens to be in the body, right? You also get the production of memory cells, which memory cells look an awful lot like the original B cell did. You just save them for later in case you get the same infection again twice. That way you can mount an immune response much faster and hopefully not get sick the next time. All right. Okay, so next thing we're going to do is zoom in just a little bit closer on some of the details of this. Right? Remember, all of this initially requires an antigen-presenting cell, right? So that is any phagocytic cell that has started digesting the pathogen and is going to go start showing it off to T cells in the immune system. And the way they do that is the antigen-presenting cell displays the antigen on a surface protein that's called an MHC, a major histocompatibility complex. And there are two different types of MHCs. If your antigen-presenting cell is using an MHC2 molecule, that means that it found something just floating around your body, so an exogenous substance right outside the cell, and that is going to stimulate helper T cells, right, CD4 T cells, and they will mature and start secreting cytokines that stimulate the rest of the immune system, like those B cells that we just talked about, right? If it happens to have an MHC1, right, like this guy, MHC1s detect endogenous or intracellular, inside the cell, antigens, right? They become CD8 positive mature T cells, right, cytotoxic T cells. They're going to go on and destroy whatever cell happens to have that antigen, because the conclusion is, if the cell has that antigen and it's foreign, there must be a virus or some other foreign pathogen that's infecting that cell. So we want to destroy it before the viruses burst out and go on infecting other tissues. All right, next thing, a little bit more on what's happening with those B cells. So remember, we had to have two things happen. The first is our B cell that has antibodies stuck in its surface. Those antibodies are different on every B cell. So each B cell has a unique antibody that recognizes a particular antigen, right? Remember, the antigen is just whatever molecule the antibody happens to recognize. They're normally proteins on the surface of bacteria or viruses. Okay? So if your B cell's antibody recognizes the antigen, and we get activation from a helper T cell that recognizes the same antigen, right? This is basically showing you that the MHC on the B cell and the T cell receptor on the helper T cell, that's the CD4 positive T cell, they recognize the exact same antigen. The T cell releases these cytokines, remember, chemical signals that stimulate the B cell. That is what is triggering division, right? So this term clonal expansion, we say that the B cell is going through clonal expansion because we don't want to just copy every B cell in the body. We want to very specifically clone that one, because this is the B cell that has the matching antibody that we need. That way, when the B cell divides and turns into these plasma cells, it's secreting the correct antibodies that match our antigen. If we had every B cell divide, we'd be making a bunch of random antigens, most of which wouldn't be specific to our pathogen at all. Okay, so once all that happens, we now have our two major cell types. Plasma cells that secrete free-floating antibodies out into the blood, right? That's these guys. and memory cells that are going to hang around for later in case a second infection comes weeks or months or years down the road. The hope is that you have a small army of those memory B cells that can divide much, much faster, amplify the immune response more faster, instead of the slow response that we got here, starting with only a very small number of cells. Okay, so that's why if you've had the exact same infection once, as long as you had a quality immune response and it hasn't been so many years or decades that the cells are gone, you shouldn't, hopefully, get sick the second time around. All right. Okay. The next video on the list is going to be going into details of how antibodies are produced and the structure of antibodies, so follow along in the playlist. It'll make more sense if you go in order.